As the climate changes, we must understand the biology of our fish stocks to better support the conservation of our global fisheries. Atlantic cod has multiple subpopulations, the most common of which are the homozygous genotypes HBI1 and HBI2. Mutations result in a significant divergence in structure that completely changes the optimal living conditions for the fish. We use JMOL to analyze how amino acid substitutions in the beta-1 chain of the Atlantic cod cause significant structural differences that leads to variation in optimal living conditions.
In order to understand the ramifications of these mutations, one must understand how hemoglobin functions. Hemoglobin is a tetrameric protein capable of binding to oxygen for the purpose of transporting it to body tissues. Hemoglobin consists of 2 beta chains and 2 alpha chains, which are folded around their respective heme groups. The molecule structure is capable of fluctuation in shape as the molecule binds with oxygen. Each heme group in a hemoglobin molecule possesses an iron cation which bonds with the oxygen. Upon a bond forming at one site, the other unbounded chains are able to shift more freely. This shift, which is able to be seen in the diagram, allows the subsequent bonds to happen more easily. This occurs once again when the molecules are deposited, as the presence of oxygen changes the molecule's shape and allows the molecules to be released efficiently. The efficiency of hemoglobin and the oxygen affinity are also affected by other factors such as temperature, pH level, the presence of carbon dioxide, and 2,3-DPG molecules.
Because of hemoglobin polymorphism, there are multiple subpopulations of Atlantic cod. The most common homozygous genotypes are HBI1 and HBI2, which have their primary differences on the beta-1 chain at the 55, 62, and 122 locations.
As shown in the diagram, the mutations in the HBI1 genotype are methionine 55, lysine 62, and leucine 122. Beginning with the properties of the amino acids, methionine is very hydrophobic and nonpolar. Lysine is hydrophilic and basic, meaning it has an electrically charged side chain. And leucine is similar to methionine, as it is very hydrophobic and nonpolar. Looking at the HBI2 genotype, the mutations on the beta-1 chain are valine 55, alanine 62, and methionine 122. Valine is very hydrophobic and nonpolar, and so is alanine. Methionine is very hydrophobic and nonpolar as well. By observing these differences, it can be inferred that the substitution at site 62 is most responsible for the differences in the folding of the beta-1 chain between the 2 genotypes, as lysine is hydrophilic, whereas alanine is hydrophobic. However, it is also important to recognize that the mutations at locations 55 and 122, while their amino acid replacements are similar, still alter the folding pattern of the beta-1 chain of each hemoglobin molecule.
Our hypothesis is that the mutations at the 55 and 62 positions are directly responsible for the functional differences between the HBI1 and HBI2 genotypes. Our first piece of evidence was a study by Colafrancesi et al. Published by Omics, a journal of integrative biology, which simulated the substitution of each valine and alanine in the HBI2 genotype with methionine and lysine, which are the respective substitutions in the HBI1 genotype. They found that the substitutions at 55 and 62, the same locations of the substitution that we see actually between the 2 genotypes, is what changed the hydrophobicity of the protein the most. We also know that hydrophobicity affects oxygen affinity, which determines what temperatures hemoglobin functions best in. Second, we have evolutionary support based on the correlation between genotype frequency and environment, so we can conclude that the mutations give a selective advantage based on oxygen affinity to each genotype in their respective environments.
In addition to the different hydrophobicity of the 2 genotypes, outside sources are also affecting the oxygen affinity of Atlantic cod hemoglobin. One such cause is rising oceanic pH levels. As the pH level of fluid in the blood changes, hemoglobin structure is altered from a taut to a relaxed state, which allows the polypeptide chains to move. After this transition, the oxygen molecule is able to attach to the hemoglobin molecule, meaning the molecule's affinity for oxygen increases. However, because Atlantic cod absorb surrounding water and thus carbonic acid into their system, the pH levels of oceans can affect the functionality of their blood. Since the oceanic pH level is becoming more acidic, the pH of the fluid in Atlantic cod blood is changed, which is a condition called acidosis. This condition is extremely dangerous to the organism and may be one of the causes of decreasing populations of Atlantic cod. Other factors that can affect the ability for hemoglobin and oxygen to bond include temperature and the presence of 2,3-DPG molecules.
It is essential for governments and fisheries to take into account the existence of different Atlantic cod hemoglobin variants while considering how to best conserve global fisheries. Since the divergence of the Atlantic cod hemoglobin types occurs along the lines of temperature, we can infer that the HBI1 type is more adaptable to the changing climate than the HBI2 type. As hypoxic regions become more expansive, this would increase pressure on the HBI1 subtype to migrate to cooler areas, as the preferred temperature of the HBI1 subtype decreases in low oxygen environments. Therefore, an increase in overall water temperatures would lead the HBI1 type to move northward to colder areas normally inhabited by the HBI2 variant, creating competition between the two cod variants. In such a scenario, the HBI1 type would have a selective advantage over the HBI2 type, causing the HBI2 variant to die off at a faster rate because there are no cooler waters for it to migrate to. The loss of habitat due to increasing temperatures, hypoxic zones, and the invasion of other cod subtypes would ultimately drive the HBI2 subtype to extinction. If that occurs, this will harm the biodiversity of the entire species, heightening the risk of extinction for the species as a whole. As seen on these maps towards the right of the screen, which predict the health of cod stocks as ocean temperatures increase by 1, 2, 3, or 4 degrees Celsius, it is the terribly overfished southernmost cod stocks, such as those of the North Sea, that are going to be the most immediately affected by increasing temperatures. With only a single degree increase in temperature, it's predicted that 2 North Sea stocks will collapse, and as the temperatures continue to rise, we can see that the southernmost North American cod stocks will also begin to decrease and collapse due to the forced northern migration of the HBI1 type. The collapse of cod stocks will greatly jeopardize the fishing industry. Therefore, it is critical to both the health of ocean ecosystems and the health of the fishing industry that governments and the fisheries better understand the biology of Atlantic cod subpopulations to avoid the overexploitation of any particular stock. That understanding will enable us to take essential, effective measures to protect threatened cod stocks by taking action against climate change and by implementing key fishing regulations to move toward more sustainable fishing practices in order to prevent the extinction of the Atlantic cod.